welcome to the live practice session for the course Applied Environmental Microbiology. Yeah, this week I was not able to handle the session on Monday, so we are having it today. So next week, like from Monday, the next Monday we'll have it, which is day after tomorrow. So thank you for joining in, and if you have any doubts from any of the previous sessions or previous live sessions as well as the lectures, you can just post it in the chat box, or you can also like unmute yourself and ask. And but before that, please confirm if uh, the screen is visible as well as my audio is clear. Is the screen visible and the audio clear? Please give a thumbs up or just a okay in the chat box. Mm. Gomati Velaragan, am I audible? And is the screen shared properly? Okay, thank you. Okay, so with that, we'll just start the session today. If you have any doubts or any clarifications, you can just post in the chat box. So the week 8 course content essentially focused on what water and wastewater microbiology, built microbiology, and exposomes. So with that, we'll just start solving some questions now. So you just have to tell me which of the following is not a common method for treating wastewater. We are starting with a very basic and a simple question. You can just choose the option and post it in the chat box. For each question, I'll just wait for a few minutes for you to answer. You can think and answer, and after that, I'll just uh, explain the answer again. thermal treatment is not a common method for treating wastewater. So when we talk about wastewater treatment, we know that it can be primary, secondary, and tertiary treatment. There are different stages of wastewater treatment as such. So in that also we have different techniques that we use for removal of different kinds of wastes or pollutants in the wastewater. So in that we have physical treatment. So in physical treatment we know that we'll be using some kind of physical forces only. There won't be any other reactions, so we'll just use physical forces. We'll apply pressure, we'll make density separation, like centrifugation and stuff, uh, ultrasonic treatments. All these fall under physical treatment category. When we talk about chemical treatment, we are entitled to perform some chemical reactions, at the end of which the pollutant is modified or mineralized. So we use different chemicals like some coagulants, and also some catalysts in some reactions. For example, antibiotic and PFAS kind of pollutants, we go for some oxidation processes. So redox reactions, we use catalysts in that, and all these come under the chemical treatment. When we talk about the biological treatment, so we know that a wide range of organics could be present in the wastewater, and they can be either treated in using aerobic or anaerobic processes, which we have seen in even previous sessions as well. So that is possible. Thermal treatment is possible, it's not like it's not possible, but we generally don't prefer thermal treatment for wastewater treatment. So when we talk about some sludge treatment after chemical or biological treatment, we have some sludge, and that goes to thermal treatment, is kind of a common stuff. But the wastewater in itself undergoing a thermal treatment is very rare, and it is also technically um, so energy consuming, money consuming, and also you need a large setup, infrastructural setup and volume to handle wastewaters using thermal treatments. They are not as effective as the other treatments as well. So that is the reason thermal treatment is generally rare. Now the next question is, which of the following is not a function of microorganisms in wastewater treatment? Again, a simple question. So we have seen different microorganisms and their role in for the past 2 weeks at least, and all of these topics is covered in there as well. Exactly. So yeah, microorganisms, they decompose organic matter. So as I told earlier, it could be aerobic process or anaerobic process. They do removal of nitrogen and phosphorus as well as heavy metals. So we have seen this in different kinds of bioremediation strategies. Whereas filtration of solids, obviously we use membranes, and that is not a biological function. Okay, so we had a conversation about um, biological treatment, thermal treatment, and stuff. So here we have some set of uh, chambers or tanks present in a typical wastewater setup. You just have to match those with the correct function of each of the chambers. These fall under different sections of a wastewater treatment unit or a plant. So you just have to match them with their respective roles. Aishwarya, were you trying to tell something or was it just a glitch? If you were trying to tell something, please feel free to unmute yourself again. Okay, I'll just wait for a couple of more minutes uh, to see if the other person would also want to answer this question. So you just have to match the wastewater treatment unit with its function. So I'll just wait for you for a couple of minutes and then discuss the answer. So the right answer is C here. So a grit chamber we generally use to remove sand and gravel. So this comes at a very early stage in the wastewater treatment unit. So when you have to treat the wastewater, you cannot assume that's only water. You will have some kind of other solid wastes also in that wastewater, which are like suspended and stuff. So out of which first you will have a screening chamber. So in that screening chamber, that essentially removes bigger particles like some plastic cups, wrappers, papers, and stuff. So these are all removed in the screening chamber, and then comes the grit chamber for the removal of sand and gravel because they are inert particles. They are also large, and also they have a higher because they have a higher density. This is kind of a settling chamber only where you give enough time for the sand and gravel to settle and then transfer the remaining wastewater to the next unit. So when we talk about this extended aeration tank, so this works well for low strength sewage. Why? Because when it is low strength sewage, the nutrients in it are very less, and microorganisms will take some time to grow in that. Like the nutrients are not very high in concentration. So you provide some extended aeration, allow more. oxygen supply, and the microbes can better perform in this condition. And hence an extended aeration tanks works well for a low strength sewage. And after that, once this microorganism has consumed the organic matter in the wastewater, there is going to be multiplication of that microorganism, so the biomass is going to increase essentially. So this increase in biomass, mm, will we will have to remove this increase in biomass from the wastewater. So there is going to be a solid liquid separation, and this biomass, along with some amount of untreated waste matter, and essentially this matrix will be held together by this EPS, which is the extracellular polysaccharide. So all this together constitutes your sludge part, and in order to separate the wastewater, kind of the treated wastewater and the sludge, you need a primary sedimentation tank. Again, here you will have a tank configuration, and you will allow the water passage in a particular flow, such that it remains in the chamber for essential amount of time for the sludge alone to get settled in that chamber. So there, your sludge alone gets settled, and you just transfer the wastewater to the next unit wherever you have to take it. And next comes this so that pairs with the removal of suspended solids. And then you have this tertiary treatment units, where you have this removal of nitrogen and phosphorus. So now you have just taken the carbon stuff, which is the organic content from the wastewater, and next you will do this nutrient removal. If you don't do the nutrient removal, again uh, it is going to be detrimental if you are going to let out the water with high nutrients. You know about eutrophication and stuff. So that is the answer for this. Match the following. Sedimentation tank and grit chamber are used for removing the solids only. So in primary sedimentation tank, you remove the suspended solids, and again you get a sludge. After the aeration tank, you will have again a sedimentation tank that is also used to remove the suspended solids. Now the suspended solids will be your grown biomass along with the EPS. Okay, next is kind of a problem question. A 100 ml wastewater sample is diluted to 500 ml in a BOD bottle using distilled water. After 5 days of incubation at 20 degrees Celsius, the initial and final dissolved oxygen concentrations were 9 milligram per liter and 3 milligram per liter respectively. So the initial concentration was 9, and after 5 days it was 3 milligram per liter. What is the BOD of the wastewater? So I have given the essential information for you to figure out the BOD of the wastewater. You just have to give me the answer. So the right answer is 30 milligram per liter here. So the reason is we have the DO initial as 9 milligram per liter and DO final as 3 milligram per liter. So DO depletion is 6 milligram per liter here, and our assumed dilution factor is 5. So we have diluted it 5 times from 100 to 500. So therefore 6 into 5 is equal to 30 milligram per liter is the BOD observed in the original wastewater. So the concept here is BOD is the biological oxygen demand, wherein we try to estimate how much amount of oxygen will be required for the organic matter to be completely depleted by uh, microorganisms through biological methods. So that is what is the biochemical oxygen demand. You also have chemical oxygen demand, which essentially tells about the amount of oxygen required to chemically oxidize the organic waste. So they both are different, and you also have a conversion factor, like assuming if this is a COD, then this could be the BOD. So that kind of trade-off you have. And also in every wastewater you should also be noticing about what is the BOD to COD ratio, because that tells you about whether the water has biologically degradable organics or what. So because if it is only going to be chemically treatable and you go for a biological system, that is also a waste of time. So you need to have a ratio of BOD to COD in the acceptable limits. So that is the significance of BOD and COD in wastewater. Coming about the estimation of BOD, what you essentially target is the estimation of the dissolved oxygen. So you have a fresh wastewater sample, you estimate the dissolved oxygen present in it. So usually there is a common standard method called Winkler's method that we use. That is a titrimetric method. Nowadays you have sensor based methods and other modern and more high throughput reliable techniques as well. But essentially you use a titration method called Winkler's method. Initially you check the dissolved oxygen, how much of dissolved oxygen is present in the water, and you put it into a BOD bottle which is completely airtight, and you incubate it in a BOD incubator. So after 5 days you come back and check the dissolved oxygen. You see how much of that dissolved oxygen is reduced. It means that that amount of dissolved oxygen was consumed by the microorganism, and the water, the organics in the wastewater has been degraded. So you can do it as BOD 5 or BOD 3. So you can run the reactions for 5 days or 3 days. It depends on the place, temperature, etc. So ideally in foreign context they did BOD 5 because of the kinetics of microorganisms, which was usually low. In Indian context you can do it as BOD 3 as well, which is you run the experiment for only 3 days. That also gives you reliable results, but you have to be consistent with the protocol that you follow. So essentially while estimating the BOD, you will just estimate the dissolved oxygen and find the difference. So this tells you what is the oxygen demand that you require. So based on this, when you go for some aerobic treatment processes, you can design your oxygen or air inlet, how much at what rate you are giving and stuff. All these you can determine. So this is how all the concepts are related. Okay, so the next question is about the tertiary treatment system. What does it involve?
ஐயையோ!
So the answer for this question is both B and C. So ideally, when we talk about wastewater treatment, we have primary, secondary, and tertiary treatment. In primary treatment, and we also saw about physical, chemical, and biological treatments that we use, right? So the primary treatment essentially involves only the physical treatment, wherein you try to use this screening chambers, grit chamber to remove all the suspended particles, and after that there comes the secondary treatment, which is mostly biological. So hereafter only you will have this biological and chemical treatments. So in secondary treatment you essentially have the biological treatment like activated sludge process, or anaerobic process, whatever. And after that, in tertiary treatment, you again go for biological or chemical techniques. So that which is essentially the removal of nutrients, like we saw about the removal of nitrogen and phosphorus, and removal of persistent contaminants. Let's say we have some antibiotics or other emerging contaminants, PFAS, microplastics, whatever that are present. You have some technique to remove them, like advanced oxidation processes and all we use in some cases. Adsorption we use, adsorption, advanced oxidation process, all of this, some amount of filtration, disinfection we use. So all these comes in the tertiary treatment. So both B and C, which is the removal of nutrients as well as the removal of persistent contaminants, both comes in the tertiary treatment of wastewater. The next question is, which of the following organisms are involved in biological phosphorus removal? Very easy question. If I have also I've given the options as well. So for this is polyphosphate accumulating organisms and glycogen accumulating organisms. So there is a trade-off between both of these organisms, and we'll just see it in just a few more slides. So I have a separate slide for that. In that, we'll just check how these. So when we talk about nitrosomonas and nitrobacter and option B and C, we obviously know it's about nitrogen cycling. And estrogenic and estrogenic bacteria and methanogenic archaea, we are talking about anaerobic digestion process, which is about methane production. So the correct answer for this is polyphosphate accumulating organism and glycogen accumulating organisms. Okay, so the next question is, what is the significance of anaerobic zone in the EBPR process? So first of all, you can just someone give what is a EBPR process in the chat box, and then you can give the correct option as well, whatever you think is the correct option in the chat boxes. Can someone give in the chat box like what is a EBPR process? Okay. So the right answer for this question is that the anaerobic zone of a EBPR uh, helps phosphorus accumulating organisms assimilate the volatile fatty acids and produce polyhydroxybutyrate storage products. So, and EBPR stands for enhanced biological phosphorus removal process. So this is basically about phosphorus removal, and here the organisms involved are the PAOs, which is phosphorus accumulating organisms. So in the EBPR process, there is going to be the cycling of this phosphorus accumulating organisms between the anaerobic and aerobic zones. So in anaerobic zone, what happens is, because there is no oxygen or nitrate present, so they take up the volatile fatty acids that are present in the system. So we know that volatile fatty acids, for example, acetate, butyrate, etc., from the wastewater. And in order to facilitate this uptake, what they have is the PHBs, which is the polyhydroxy butyrates. So it comes from, so the organism tries to convert this VFAs into polyhydroxybutyrate compounds. Okay, so for this they use the polyphosphate. So in the anaerobic zone, volatile fatty acids are stored in the form of polyhydroxybutyrate. That metabolism happens in the anaerobic zone in a enhanced biological phosphorus removal system. Which of the following is not considered as a part of an exposome? So exposomes is generally about how an organism is exposed to the environment. So in that we have, it's the interaction of the organism with chemicals, biological agents, as well as physical factors. But genetic factors we will not consider, because the exposure during the lifetime of an organism is only the factor that we look into when we talk about exposomes. Okay, from the given statements, you have to find out which of the following is or are true and false. You have to basically state whether both the statements are true, both are false, or which is true and which is false. So you have to select that. Statement A is biomarkers could be used to assess exposure to environmental toxins or other environmental factors. They can be measured in various biological samples such as blood, urine, or saliva. Statement B is by measuring the levels of certain biomarkers in the body, researchers can assess the extent of exposure to various environmental factors and evaluate the risk of developing a disease. what could whether the statements are true or false, you can just post it in the chat box. Okay, so the right answer for this is both the statements are true. So generally we use biomarkers to assess the environmental exposure. Like when we are exposed to some kind of toxins or chemicals, there are certain characteristic biomarkers that we could find from biological samples like blood, urine, and saliva. And the quantitative measurement of those biomarkers can help us assess the extent of which the exposure is happened. So if the exposure is intense, then the biomarker presence would also be proportionally high. Both qualitative and quantitative assessment of exposure to pollutants and other environmental factors can be done using biomarkers. Biomarkers are nothing but some characteristic biochemicals that you would be testing from your sample, which has been produced in response to the exposure. Okay, one very easy question. Which is the primary building block of proteins? I guess you can answer, all of you can answer this question, and you won't need much time for this. Exactly. So amino acids is the right answer. Okay, so the primary objective of secondary wastewater treatment, I think we have seen this extensively. Which of the following is the primary objective of secondary wastewater treatment? Okay, so the objective of secondary wastewater treatment is BOD removal. So, primary treatment, it is about suspended particles and physical treatment. Secondary treatment is biological treatment, so it is about organics and BOD removal. And then comes the nutrient removal and pathogen removal. Pathogen removal, you finally go for disinfection process, and that is kind of a pathogen removal process. Okay. What kind of a system is a trickling filter? Exactly. So, trickling filter is a biofilm system. So, trickling filters belong to a category where we call it as a biological system which supports attached growth processes. So, you have a kind of a media over which the microorganisms will be present in the form of a biofilm, and they are going to be present in the treatment system, and water is going to be passed. So, the microorganisms that are coated over this media uh, feed on the organics in the wastewater, and similarly for aerobic processes you also supply enough oxygen in that process. When we talk about suspended growth systems, general process like activated sludge process where you directly have microorganism in the wastewater itself is a suspended growth system. And obviously trickling filters are not anaerobic, they are chemical reactors, biochemical reactors in one sense, but they are more a biofilm reactor. So you can assume that there will be a media over which there will be a coating of microorganism, and this you call as a biofilm, and the microorganisms from this matrix try to uh, perform the organic degradation. Again, a repetitive question. Primary treatment removes which of the following? You just have to be careful with the terms that you deal with. I'll just give you suspended solids. So we use settling chambers, a grid chamber screens to remove the suspended solids. Okay. How does a plug flow reactor differ from a CSTR? CSTR is continuously stirred tank reactors. So this is a very open-ended question. Based on whatever perspective you find the difference, you can just post it. concentration of the pollutant is going to be different here. Okay, which is going to be less here. So essentially at this point it will have a even lower concentration, and based on whatever is your treatment efficiency, your wastewater will again come out here. Okay, uh, the concentration here, here and here, I mean at any particular point in the plug flow reactor is going to be different, but at this point it is going to be uniform. That is what we assume, because there is no uh, mixing here. So you call that mixing as a axial mixing, which is the mixing in the direction of flow. There is no mixing in the direction of flow. Okay, and this each part you call it as plugs, and this is what entirely you call it as a plug flow reactor. Each of this plug is going to be having a uniform concentration, but the concentration of the pollutant is not uniform throughout the reactor. Whereas when you talk about the CSTR, you are initially feeding the CSTR with some wastewater, and there is perfect mixing, and your even the microorganisms will be present here in this system. They will take up the organics, and at any given point the uh, concentration of the composition is going to be uniform inside a CSTR. So this concentration difference is the key between the PFR and CSTR, and that arises from the factor that there is no axial mixing in PFR, whereas there is mixing in the CSTR. So that is the key difference between both of these reactors. The mixing pattern, the mixing characteristics is the key difference between a PFR and a CSTR. I hope it was clear. If there are any doubts, please post in the chat box. Okay. Which process do we generally use for sludge digestion? Right. We generally go for anaerobic process. So what we essentially have to do is sludge stabilization. So the sludge from the treatment process or aerobic process is going to be highly active, and that is why you again use it and call it as activated sludge process. You have to stabilize the sludge. You have to remove the nutrients, organics and stuff. So you generally go for a anaerobic process, which will convert this into methane and other gases. Okay, next is the assertion and reason question. So the assertion is activated sludge reduces the treatment time. The reason is the biomass concentration increases the reaction rate. You have to tell which of the statement is true. If either of it is true or false, you have to just mention. So both the assertion and reason are correct. Activated sludge reduces the treatment time because the microorganisms are already in the log phase. They are like in the peak of their growth. Okay, so when you are adding them to a wastewater system, they'll happily survive and they don't have this lag phase and stuff. So the treatment time will reduce. The kinetics is increasing and the treatment time is reducing. So that is what is the reason again. Reaction rate or the kinetics is getting increased. So both the statements are right. So out of these given statements, identify which statement is false. The first is exposome includes lifelong exposure. Genetics fully explains chronic disease. Single time exposure studies are sufficient, and omics technologies help exposome studies. Which of the following statement is or are false? Just give the option uh, in the chat box. Okay, so the false statement here are B and C. So, coming from the statement A, exposomes is the measure of all the exposure of an individual in its lifetime. So, it includes the lifelong exposure. While when we talk about genetics, genetics plays a role, but it is not like fully accountable for chronic diseases. Other environmental factors also contribute to explain chronic diseases. And single time sufficient studies are not enough, because the exposome is a lifelong concept and there is going to be temporal variation, the variation with the time in the exposure. So therefore a single time exposure study is not sufficient, and omics technologies is essentially used to in the exposome study. So when we are exposing a microorganism to some kind of conditions and checking its genomics, proteomics, transcriptomics, metabolomics, etc., then we can integrate all the results and then see what exactly is happening in the system. Okay, so you have to fill in the blanks. Just give, if you know the answer, just give A and what is the answer that could fill in the blank. exposome complements dash brain gut axis is dash serotonin is derived from what, and omics technology that particularly helps in exposome is what? Okay, so exposome complements genome, and brain gut axis is bidirectional. It's not like one directs the other only, it is bidirectional uh, interaction. And serotonin is derived from the amino acid called tryptophan, and the omics study that is used in exposome is metabolomics. Basically, that is used. So, with that, we have come to the end of the question and answer. I'll just go through in the remaining time, I'll just go through these different terms. Okay, so first is crown corrosion. So, when we talk about crown corrosion, it is the deterioration of the upper portion of sewer pipes. So, as you can see in this image, there is a sewer pipe and there is going to be deterioration in the upper part, and this is going to be a microbially induced corrosion. The mechanism here is generally sulfate reducing bacteria produce hydrogen sulfide gas. So, hydrogen sulfide is a corrosive gas, and this is produced by microorganisms under anaerobic condition. So, because this is a gas, this diffuses to the pipe, top of the pipe, and sulfur oxidizing bacteria convert this hydrogen sulfide to sulfuric acid. You all know sulfuric acid again is corrosive. Okay, this acid attacks the concrete, the top layer of the concrete, and causes corrosion. So this is what you call as a crown corrosion. It takes in the top of the pipe through which you supply the wastewater or you supply the water inside your piping system basically. So the next term is patina type 1 and type 2. So patina is a surface layer that is formed in the materials due to environmental exposure. So in type 1 patina, what happens is it is going to be thin, uniform and adherent, and it reduces further corrosion. For example, it's like a stable oxide layer on metals, and this is kind of a protective layer. When you talk about type 2 patina, it is a non-protective, uh, unlike the first one, it's going to be porous, uneven, and flaky, and this will like promote the continuous corrosion of the material and finally leads to the degradation of the material. So the next term is the emerging contaminants. So emerging contaminants, as in they are also pollutants, but they are very recently identified as pollutants themselves, like microplastics or PFAS. So you know that they are pollutants, they cause serious harms, but they are more newly recognized, and as a result of which we don't have any regulations to control them. Like we don't know, when we talk about this allowed amount of nitrogen in wastewater or phosphorus in wastewater, or any other metal in wastewater, you have a permissible limit. You have different organizations that tell you this is the permissible limit. Even for pathogens or coliforms, you have this permissible limits. Okay, whereas for this emerging contaminants, though you know they are contaminants, we still don't have regulations telling how much amount is allowed or how much is permissible in water systems. So that's why they are known as emerging contaminants. So examples include pharmaceuticals, hormones, personal care products, and microplastics. uh, these are biologically not very active and are present in trace amounts, and these can also be degraded by co-metabolism. Now only we have discovered that it is a pollutant, so people will be actively working on finding what kind of toxicity they are causing, at what concentrations they are causing, and other group will be dealing with how to, like how their transmission is or transport in the nature is working, and few other researchers could work on different methods of degrading this physical, chemical, biological, whatever methods, thermal methods even, for degradation of this contaminant. So these newly identified contaminants are called as emerging contaminants. Okay. Okay. This we have seen in detail already. The heading is wrong anyways. So here we are talking about the enhanced biological phosphorus removal system. So we have this phosphorus accumulating organism that store phosphorus as polyphosphate. So in the anaerobic phase, they uptake the VFAs and form PHBs, polyhydroxybutates, and they release phosphate. So then in the next phase, what happens is, in the anaerobic phase, sorry, in the aerobic phase, they take up the phosphate and store as polyphosphate. And finally, this microorganisms you take out and the sludge is wasted, through which you remove the phosphorus from the system. Primary clarifier and secondary clarifier, this I told in the very first slide here. So primary clarifier is usually used after a screening to remove the settleable particles like sand and gravel. So this is a physical treatment, and it's also a primary treatment. Secondary clarifier, when you talk about, it comes after the biological treatment. So once after the biological treatment, you will have more number of biomass, which you call as sludge, and you want that to be separated from your water, which is a solid liquid separation. So here you separate the biological flux or biomass and have the sludge, which is the activated sludge in your activated sludge process, and you recycle it back to the system. So this is the difference between primary and secondary clarifier. And this also we have seen what is the difference between COD and BOD. So COD measures the oxygen required for chemical oxidation, whereas for biological oxidation you call it BOD. And we just saw the difference between a plug flow reactor and a CSTR, and you also have a completely mixed flow reactor. This is also similar to the CSTR, but yeah, you can operate that in batch as well, so it depends on the mode of operation as well. And in sequential batch reactors, which I think we saw in one of the previous sessions as well, there will be sequential like let's say four sequential reactors. First you will fill one, allow it to react, and then you transfer it to the next stage for settling. So in the meantime, you will fill the first one, reaction will happen in the second one, settling will happen in the third one, and in the fourth one, decantation will happen. So you have four different reactors that are present together and are operating simultaneously, but you transfer the wastewater from one system to other for each of this step to happen, which is filling, reaction, settling, and decantation, and that is why you call it as a sequential batch reactor. And next, the last few terms are fog, which is generally abbreviated as fog. It is fat oil and grease. It is found in domestic wastewater. the lipid content basically. This causes clogging. You all know it is very difficult for microorganisms to degrade this, and also they are not dissolved also, so they cause a lot of problem like clogging, scum formation, and other operational issues. And another abbreviation that we generally use is the ARG, which is the antibiotic resistant genes, and the ARMs, antibiotic resistant microorganisms as well. So when we talk about antibiotic resistance, what happens is when microorganisms are continuously exposed to some antibiotics, they develop a genetic power that helps them to resist the antibiotic action. So why do we generally use antibiotics? These antibiotics are substances produced by one microorganism against another set of microorganisms. So these we use this antibiotics in terms of medicines. When we have some infections, bacterial or viral infections, we use this antibiotics as medicines. And when these are like generally let off, what happens is the microorganisms that are present in the environment can freely interact with this antibiotics, and they try to develop resistance against these antibiotics. So the result of it is, when we are going to be infected with this resistant microorganisms, even though we take antibiotics, our disease will not be cured, and we need to constantly find some other antibiotics that could work. So this is a serious loop from microorganisms to animals to even the extent of plants and humans. This is a serious deal. Okay, and there is a lot of research going on in this field as well. And last topic is about the adductomics. So adductomics is a study of covalent adducts formed between reactive chemicals and biomolecules. You just study the interaction here, and this helps to assess internal exposome, like how the exposure to different conditions inside the microorganisms itself creates some kind of bonding between the biomolecules that is present in the system. So this is all for today. Thank you for joining in. If you have any doubts, please post them in the chat box. Otherwise, we'll meet again on coming Monday. So this class was just postponed from the Monday to today. We'll, as usual, meet on coming Monday. Any doubts? If you have any doubts, please feel free to post in the chat box.